Moving inside the cell, the major component we have is the cytoplasm. This is the semi-fluid medium inside the cell. It's composed of a lot of water, salts, and dissolved organic molecules. It's jelly-like. It's about 75 to 90% water. And as we saw from Unit 2, there's a lot of reasons that cells would want a large portion of water due to its polarity and other chemical properties. This is the site for various metabolic reactions. Within the cytoplasm is the cytoskeleton, also known as the microtrabecular lattice. It consists in part of microtubules, which are slender protein tubes. These help maintain cell shape, and they assist in the movement of cell and organelles. Perhaps contrary to our concept of a skeleton, the cytoskeleton is dynamic. That is, it's always changing. It's assembled and disassembled as needed. Within the cytoplasm are different organelles. Looking at the organelles of eukaryotic cells, the term organelle originally referred to only membranous structures, meaning those that were surrounded by some type of membrane. It now refers to any well-defined cell structure that performs a particular function. The cell is compared to a factory, where raw materials enter and different departments turn them into various products. So besides taking this raw material, doing something with it, turning it into a product, we also have waste that's produced, and we have to get rid of that. Let's start with the endomembrane system. This consists of the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, and several vesicles. Vesicles are tiny membranous sacs. The endomembrane system as a whole acts as the transportation and product processing section of the cell. It compartmentalizes the cell to make it more efficient so that enzymatic reaction is restricted to specific cell sections. We looked at the nuclear envelope earlier. Now let's look at the endoplasmic reticulum. Here's the nuclear envelope here, and we can see how this network of membranes is continuous with that. The endoplasmic reticulum is a network of interconnected membranes. We can see here that there's two distinct types. Here we see one section that has tiny spheres on it, right? So we're looking here on a micrograph. We can see that one looks rougher than the other one. That's why we call this the rough endoplasmic reticulum, or you'll see this abbreviated as the rough ER. The other section does not have ribosomes on it and looks a little smoother, and so we call it the smooth endoplasmic reticulum. Because the rough ER is studded with ribosomes, ribosomes are protein factories, and so this is involved in the processing, folding, and modification of proteins. The smooth ER does not have attached ribosomes. Instead, this is where phospholipids and steroids are made. It also stores calcium ions, and depending on the cell type, it can have various other functions. Ribosomes, we just stated, are the site of protein synthesis. They use messenger RNA, denoted as mRNA, as a template. We'll see this later. Ribosomes are made up of two subunits. You have a large and small subunit. The subunits themselves are made up of ribosomal RNA, that's denoted rRNA, and protein molecules. The ribo refers to ribonucleic acid, which is RNA. Somes refer to bodies. Ribosomes are found individually or in groups of polyribosomes, poly meaning many. With a polyribosome, several ribosomes are associated with a single mRNA. Ribosomes are also attached to the rough endoplasmic reticulum, as we just saw. They can also be free in the cytoplasm. Flipping back to our diagram of an animal cell, we can see ribosomes here, as well as free either in single units or as a polyribosome within the cell. The Golgi apparatus is so named for the man who discovered it. It consists of a stack of 3 to 20 slightly curved sacs, kind of like, looks like a splat or a stack of pancakes. They are usually found near the nucleus. They are referred to as the shipping center or post office of the cell. In animal cells, one side is directed toward the endoplasmic reticulum, and the other side is directed toward the plasma membrane. It's referred to as the shipping center or post office of the cell because the Golgi apparatus collects, sorts, packages, and distributes materials such as proteins and lipids within the cell or out of the cell. The Golgi apparatus receives proteins and also lipid-filled vesicles that bud from the endoplasmic reticulum. We just saw that the rough ER is involved in modification of proteins, and the smooth ER can be involved in manufacture of things like lipids. So it can receive that from the rough and smooth endoplasmic reticulum, and proteins that are made in the rough ER have tags or zip codes that help let the Golgi apparatus know where to send them. Lipids and proteins are modified in transit through the Golgi before being repackaged into secretory vesicles. These contents are then discharged out of the cell by secretion. So looking at the nucleus here and the endoplasmic reticulum, with the rough endoplasmic reticulum, this is where proteins are folded and processed and then packaged into vesicles, these little membranous sacs that can transport items throughout the cell. As these transport vesicles shuttle proteins to various locations such as the Golgi apparatus, it can fuse with it, and then the Golgi apparatus is going to further modify lipids and proteins, sort them, and package them in other secretory vesicles. And if something's a secretory vesicle, that means it's going to be secreted. So, as part of secretion, this vesicle fuses with the plasma membrane and is able to discharge that contents out of the cell. Similarly, if you had lipids coming from the smooth endoplasmic reticulum, then transport vesicles can shuttle these lipids to places such as the Golgi apparatus, and similarly, the Golgi apparatus can further modify them, sort, package them, and send them to different parts of the cell or out of the cell. On that diagram, we also saw lysosomes. Lysosomes are membrane-enclosed vesicles formed by the Golgi apparatus. They contain hydrolytic digestive enzymes. They act as the garbage disposal or cleanup crew or recycle center of the cell because they break down unwanted foreign substances or worn-out cell parts. They cause things to lyse or break apart. They also bring macromolecules into the cell. So here, if we have an incoming vesicle that's bringing a substance into the cell, this vesicle can fuse with a lysosome that's been produced by the Golgi apparatus, and the lysosome contains digestive enzymes that are going to break down worn-out cell parts or substances entering the cell. Vacuoles are large membranous sacs that are used to store substances such as water, sugars, salts, pigments, and toxins within cells. So differences between vacuoles and vesicles. Vacuoles are going to be larger, and they're typically going to be for storage more than transport. They're going to be more prominent in plant cells. Here we see the central vacuole, kind of the largest portion of this cell. The central vacuole provides added support as well as storage. Whenever you see plants that are wilted, it's lost some water. Whenever they perk back up, the vacuole is able to store water. Peroxisomes are similar to lysosomes. They are also membrane-bound vesicles containing enzymes. Peroxisomes break down fatty acids and amino acids to hydrogen peroxide, H2O2, which is toxic to cells. And here's where you get that name, peroxisome. However, hydrogen peroxide is quickly broken down into water and oxygen by the enzyme catalase, and water and oxygen are then harmless.